Hello, everyone. My name is Jerome Del Rosario, and alongside my group mates Elizabeth Aventajado, Joshua Garcia, Hannah Patricia Marantan, and Kaida Bernice Santiago, we will be focusing on the invasive flora and its effects to the nitrogen and sulfur cycle, alongside epigenetics and anthropogenetic effects.
The nitrogen cycle. Did you know that nitrogen moves through different environmental systems in a fascinating cycle, transferring into various chemical forms along the way? This biogeochemical process is essential for the health and sustainability of our atmosphere, land, and oceans. The cycle of nitrogen has five parts. The nitrogen fixation. The passage elucidates the principal mechanism by which nitrogen is assimilated into terrestrial ecosystems, spotlighting the critical limitation that plants cannot directly harness atmospheric nitrogen, which is a major component of Earth's atmosphere. It accentuates the indispensable role of nitrogen-fixing bacteria, either existing freely in the soil or in the symbiotic relationship with the roots of specific leguminous plants, in converting inert atmospheric nitrogen into biologically accessible forms such as ammonia and nitrate. This symbiotic relationship is underscored as being particularly beneficial in environments lacking in nitrogen, offering a vital nutrient source that significantly enhances the growth and productivity of terrestrial flora, thereby showcasing nitrogen-fixing bacteria as fundamental to the sustainability and vitality of terrestrial ecosystem. Nitrification. Upon the introduction of ammonia into the soil via the process of fixation, it undergoes transformation by nitrifying bacteria, initially converting to nitrite to subsequently to nitrate. This conversion is crucial as the nitrate represents a form of nitrogen that is highly soluble, facilitating its absorption by plant roots with relative ease. The significance of this process cannot be overstated, as nitrates serve as a fundamental component of nitrogen utilized by plants to synthesize essential biomolecules such as proteins and nucleic acids. Thus, the sequence of biochemical reactions play a pivotal role in the sustenance of plant life, underpinning the synthesis of vital organic compounds. Assimilation. Plants utilize their root systems to absorb nitrate and ammonia from the soil. These forms of nitrogen are then assimilated into the plant's tissues of amino acids, which serve as the fundamental building blocks for proteins. Furthermore, they are crucial for producing other organic compounds vital for plant growth and development, including enzymes, chlorophyll, and DNA. Mineralization. In natural ecosystems, when plants and the other organisms pass away, decomposing bacteria and fungi in the soil convert the organic nitrogen within their bodies back into ammonia. This crucial process effectively recycles as the nitrogen, rendering it available once more for other plants to utilize. The death and decay of plants matter play a significant role in contributing to the nutrient pool accessible for other plants, making the cycle especially vital. Denitrification: the conversion of nitrate back into gaseous nitrogen by bacteria can result in a depletion of nitrogen in the soil, thereby limiting its accessible to plants. Nevertheless, this process is absolutely essential for averting the accumulation of nitrate in soils and groundwater. Such as buildup can prove determ- Such buildup can prove detrimental to ecosystems and human health alike. In summary, the nitrogen cycles play a crucial role in preserving the equilibrium and accessibility of nitrogen in the environment, thereby facilitating optimum plant growth. However, human activities such as excessive fertilizer use, deforestation, and pollution can disrupt the cycle, resulting in nitrogen imbalances that can adversely impact health, growth, and ultimately the entire ecosystem.
In the intricate web of life, where every element plays a vital role, the sulfur cycle stands as a crucial thread weaving through ecosystems. Sulfur, a fundamental component of nature, enters the atmosphere through natural phenomena such as volcanic eruptions and human activities like industrial processes and the burning of fossil fuels. Once in the atmosphere, sulfur compounds undergo transformations, leading to formation of sulfuric acid and contributing to the phenomenon known as acid rain. Through deposition and washout, sulfur compounds return to Earth's surface, contaminating water bodies and soil. In the hydrosphere, sulfur exists in many forms, sustaining aquatic life, while also posing challenges in the form of pollution. In the soil, sulfur compounds undergo transformations mediated by microbial activity, cycling through mineralization and oxidation or reduction reactions. Plants play a pivotal role in the sulfur cycle, absorbing sulfur from the soil and incorporating it into organic compounds. Yet, human impacts have disrupted this delicate balance with excessive sulfur emissions, leading to air pollution and ecosystem degradation. Efforts to mitigate sulfur pollution include regulatory measures, technological innovations, and the transition to cleaner energy sources. Understanding the dynamics of sulfur cycle is essential for preventing the health of our planet and ensuring a sustainable future for generations to come.
Le Chatelier's principle is a fundamental concept in chemistry, illuminating how systems respond to external changes to maintain equilibrium. It provides invaluable insights into chemical reactions and the establishment of stable equilibriums, critical for natural processes. When applied to human impacts, it reveals the intricate connections between our actions and chemical equilibriums in the environment. Anthropogenic activities such as industrial processes and deforestation often disrupt delicate balances in the natural systems, leading to environmental degradation. Therefore, it's crucial to scrutinize our actions and assess their consequences meticulously. By understanding the repercussions of human interventions and chemical equilibriums, we can develop informed strategies to preserve the environment and biodiversity. Furthermore, Lachatelier's principle guides sustainability efforts by informing tailored approaches to mitigate human impacts on chemical equilibriums. This knowledge is essential for fostering sustainable practices and ensuring the utilization of resources. By embracing this principle, we can navigate the relationship between human activities and environmental sustainability, striving for harmony and resilience in our interactions with nature.
What are invasive species? Invasive species are non-native species that deal a great amount of harm to an environment due to it competing with the native species of that area. What counts as an invasive species? For a species to be considered invasive, they should meet the following criteria. Overall, they need to be uncontrollable. They need to be able to adapt quickly to the environment they are introduced in. And they need to be able to multiply quickly. Meeting such criteria makes them an invasive species because they are harmful for the ecosystem that they are introduced in, since they usually outperform and kill off the native species of that area. Here are some examples of invasive species of flora. We have the common water hyacinth that multiplies so rapidly that they usually densely populate the surface of a body of water. This leads to sunlight not reaching the depths of that body of water, thus submerged plants and fish are unable to receive sunlight. Then we have what we call here in the Philippines as hagonoy, which is a rapidly growing weed that is toxic to farm animals. We also have the large leaf lantana, which is also toxic and it grows and spreads fast. How does invasive flora affect the nitrogen and sulfur cycle? First, for the nitrogen cycle, there have been researches that indicate and found that invasive flora usually increase the amount of nitrogen in the soil of that area. This leads to an overall increase in the nitrogen of that ecosystem, which usually kills off sensitive plants, since too much nitrogen is actually bad for the plants. As for the sulfur cycle, while invasive flora does not have an actual effect on the cycle itself, the cycle greatly benefits the invasive flora. Since sulfur is an important nutrient for the growth of plants, and considering that invasive flora usually are rap- invasive flora usually have a rapid growth rate, invasive flora are able to take full advantage of the sulfur in the soil, thus leaving the native species of that area unable to fully utilize the sulfur in their ecosystem. It spreads fast.
The epigenetics of the flora. In the intricate tapestry of nature, plants possesses a silent but powerful language of genetic regulation known as epigenetics. Let's delve into the fascinating world of plant epigenetics and uncover its profound implications for environmental research and sustainable practices. So, first of all, what is epigenetics? So, epigenetics is a cornerstone of contemporary biological research that explores heritable changes in gene expression beyond DNA sequences. Within the realm of plant biology, epigenetic mechanisms serve as a pivotal orchestrator of gene expression and phenotypic plasticity, allowing plants to thrive amidst diverse environmental challenges. Understanding the intricacies of plant epigenetics offers a profound insight into the dynamic interplay between plants and their surroundings. By deciphering the molecular underpinnings of environmental adaptation, researchers paved the way for innovative strategies in conservation, sustainable agriculture, and ecosystem management. Here are the three key epigenetic mechanisms. And first of all, we have DNA methylation. So DNA methylation is a crucial epigenetic process where methyl groups are added to specific parts of DNA, particularly in cysteine residues. In plants, this modification mainly occurs in three specific sequences: CG, CHG, and CHH. The establishment and maintenance of these methylation patterns are carefully regulated by enzymes called DNA methyltransferases, methyltransferases, and demethylases. Sorry. Interestingly, environmental factors such as temperature changes, light exposure, nutrient levels, and interactions with other organisms can trigger changes in DNA methylation patterns. For example, extended exposure to cold temperatures can lead to widespread changes in DNA methylation, regulating genes that are essential for adapting to cold conditions and stress. This adaptability highlights how plants use epigenetic mechanisms to adjust gene expression in response to their environment. Secondly, we have histone modification. So histone modification is which are chemical changes to histone proteins are crucial for shaping how DNA is packaged and genes are controlled in plants. There are various types of these modifications like acetylation, methylation, phosphorylation, ubiquitination, and sumylation, forming a complex code that regulates gene activity. Interestingly, these modifications can work together or against each other to control gene expression. Like, for example, histone as acetylation usually turns genes on, while histone methylation can either activate or silence genes, depending on specific factors, of course. And environmental factors like drought, salinity, pathogens, and changes in light quality can trigger changes in these histone modifications, reshaping how genes are expressed. So, this complex interactions show how plants adjust their gene activity to cope with different environments. And lastly, we have small RNA pathways. Small RNA molecules, including microRNAs and small interfering RNAs, serves as key regulators of post-transcriptional gene expression and defense mechanisms against invasive genetic elements in plants. MicroRNAs derived from indigenous hairpin precursors guide the RNA-induced silencing complex to target complementary mRNAs, mRNAs for cleavage or translational repression, thus fine-tuning gene expression levels. On the other hand, small interfering RNAs generated during RNA interference pathways trigger RNA degradation or chromatin modification at target loci, leading to gene silencing. These small RNA pathways play integral roles in diverse biological processes in plants, including development, stress responses, nutrient homeostasis, and symbiotic interactions with microbes. By intricately regulating gene expressions and defending against genetic invaders, plants ensure their survival and ecological fitness in ever changing environments. Together, these epigenetic mechanisms create an intricate system, allowing plants to adjust, flourish, and peacefully coexist with their surroundings. Studying the complexities of plant epigenetics offers valuable insights into nature's remarkable methods of adaptation and endurance. As we wrap up our examination of plant epigenetics, we're reminded of the deep interconnectedness among all living beings and their environment. By deciphering nature's genetic code, we pave the path towards a sustainable future where humanity and plant life thrive in harmony. Thank you.